Hello, my dear friends. I'm Dr. Vaishali Bharambe, Professor Anatomy. If you're a student who finds drawing of histology diagrams difficult, or if you sometimes don't even understand the explanations and corrections given by your teacher, then this video is for you. Let's begin to understand how to draw diagrams of muscle. What are the slides we're going to discuss? Skeletal muscle, cardiac muscle, and smooth muscle slides. Let's start with the skeletal muscle. First thing, you should know where it is found. You should know the examples, all the different skeletal biceps and triceps and everything is a skeletal muscle, right? Then we will be discussing TS, LS and sarcomere now. Let's begin to discuss first the transverse section of a skeletal muscle. So take your color pencils and begin to draw with me. First, draw the connective tissue sheet that holds the skeletal muscle cells together. Okay. Now begin to draw transverse sections of the skeletal muscle myocyte. Remember, they get sort of pressed against each other and there will always be a good number of blood capillaries in between. So remember to draw those as well. Let's now color the myocytes. Okay. And remember to draw the epithelium of capillaries. Draw the nucleus flattened towards the periphery. Okay. Remember to draw also the connective tissue surrounding, supporting it. So see how I'm gently staining the background. You have to draw it. The muscle cells are not going to fly around in the air. So that has to be drawn as well. Let's now label this. Okay. So this was a muscle fascicle. Groups of muscle cells form a fascicle, which is enclosed by connective tissue sheath called perimysium. This is a single muscle cell or a myocyte enclosed by connective tissue sheath called endomysium. Observe how the nucleus is flattened here and lot of blood capillaries. So what should you remember? Epimysium, perimysium, endomysium. Let's zoom in a little bit more and understand further. In one corner of your diagram, start to draw the zoomed in image. Okay, these are larger skeletal muscle myocytes, only appearing larger because it's a zoomed in image. But now through the muscle cell, you're going to draw such tiny little dots. Keep drawing them, have patience. Remember to draw the nucleus flattened and towards the periphery. Epithelium of capillary, never forget, and never forget the connective tissue. Now let's label our diagram. So the muscle fascicle, blood capillary, we already knew. Here's a myocyte and covered by endomysium. You can see the flattened nucleus, and this is the myofibril showing tiny dots. So one muscle myocyte is showing all these myofibrils packed in the form of tiny little dots. Some books call it a stippled appearance. Okay. Let's now talk about the longitudinal section. Same muscle in the form of LS. What will it look like? Again, take your pencil and begin to draw such longitudinal sections. Okay. Initially, always draw very light in color because then otherwise if you make a mistake, the whole diagram becomes very messy. Stain, there are only 4 myocytes here. Stain them properly. Make sure that you draw the connective tissue also by darkening the edges. Now begin to draw such bands. These are light and dark bands which are always seen. That's why we call it a striated muscle, right? So draw these striations. Have patience. Don't get bored. Draw the flattened nucleus towards the periphery. Remember, because of the myofibril content, nuclei are flattened and pushed to the periphery. But in between, you will find a lot of capillaries which are present in the connective tissue sheath. Please remember to draw the nuclei of the fibroblasts which are present in the connective tissue sheath. Stain the cell well. There. Let's now label this diagram. These are the myocytes. the alternating light and dark bands giving the typical striated appearance. This is the flattened nucleus towards the periphery. This is the endomysium, the fibroblast, and the blood vessels, which are of course there to supply energy to the skeletal muscle for its contraction. Let's now zoom in on a drawing of a sarcomere. This is where most of you find it a little difficult. It's very easy. Okay, take the requisite color pencils, get ready. Draw two such horizontal lines indicating the myofibril. In the center, one single line, and such thick filaments, the myosin filaments. Here are the two Z lines, and attached to them, remember to draw the thin filaments, the actin filaments. Okay? Let's label what we have drawn now. So, thick filaments, myosin filaments, thin filaments, actin filaments. A part of it that looks dark called the A band, a part of the myofibril that looks light called the I band. In the A band, you will find a lighter zone called the H zone, where only the filaments are there, in the center of which there is a single line called the M line. In the light band, I band, you find a single line called the Z line, here too. And the distance between two Z lines is called as a sarcomere. Okay. Now with this knowledge, let's move on to the next little diagram that you should be drawing, which is contracted sarcomere. So draw the same two brown lines, the line in the center, the myosin filaments, which are of course thick. And now remember to draw the two Z lines quite close together. Thus the actin filaments, which are thin, have interdigitated with the myosin filaments, reducing the actual length of the sarcomere. Let's label this now. This is what you have drawn. These are the two Z lines which have come closer together, and therefore the sarcomere has contracted. This is the diagram of a contracted sarcomere. Okay, let's now move on to the cardiac muscle. Cardiac muscle is striated voluntary. Remember, where is it found? It's found in the heart and sometimes a little bit in the proximal end of the pulmonary trunk. Let's begin to draw a diagram of cardiac muscle. Let's draw a diagram of cardiac muscle. So draw such somewhat branching looking cells. Color them. I tend to color my cardiac muscle a little light and dark because that's how it appears to me when I observe it under the microscope. So many times your diagram should be influenced by what you are seeing. Remember to draw a rounded nuclei. Okay, it is not a multinucleated cell. Now you will see that around each nucleus there is a pallor. There is a pale zone around each nucleus. This is called perinuclear pallor. Now let's begin to divide what you're seeing into individual cells. For this you have to draw these lines which are called as intercalated discs. These discs are indicating how myocytes are attached to each other to form a network or a syncytium. Now we have to draw the surrounding background connective tissue. Okay. Remember always to work on the nuclei a little bit because they tend to stand out well. In between these cells, of course, you will find plenty of connective tissue and blood capillaries. Draw them. Define your cells quite well. And there, let's label our diagram. So this is what you have drawn. These are the myocytes which appear to be branching, connected to each other with the help of intercalated disc, which should be drawn as shown. Each cell has a rounded nucleus with perinuclear pallor. You will be able to see such striations which are not very well defined. Okay, so draw them ill-defined compared to your skeletal muscle. The cardiac muscle itself will be found enclosed in endomysium. To remember to draw your connective tissue properly. Lot of blood capillaries, of course, we are dealing with a cardiac muscle. So I'm going to give you a ready chart comparing the skeletal and cardiac muscles. You can pause the video and read this as well. We'll now move on to the smooth muscle. Okay, so let's draw the smooth muscle. Again, here also you have to know that it's a non-striated involuntary muscle. Where is it found? In the walls of blood vessels, in the walls of intestines, in the wall of ureter, everywhere. You should know where you find your smooth muscle. Examples. And then now we'll study the LS of smooth muscle and the TS of smooth muscle. Right. So begin to draw the last two diagrams. Remember, your smooth muscle cell is spindle-shaped. Okay, so that the thick part of one is placed next to the thin part of the other. These cells are often placed in small fascicular groups. The nuclei of these cells are somewhat cigar-shaped. So draw them that way. They are all enclosed in a connective tissue sheath. So make sure you remember to draw your connective tissue. The blood vessels will be found in the connective tissue. So these are all the connective tissue sheath which are enclosing your smooth muscle, forming smooth muscle bundles or fascicles. Let's label our diagram now. These are the myocytes. This is the connective tissue sheath enclosing it. A cigar-shaped nucleus and blood capillaries in the connective tissue sheath. We'll now go on to the last diagram, which is transverse section of a smooth muscle. Remember, now you have taken a section of the LS. So wherever your section has gone through a tapering end of the smooth muscle, the section will be smaller. Where it's gone through the center of a spindle-shaped smooth muscle, your section will be larger. So the sections of smooth muscles will be found large and small, as you can see. Color them up. The nucleus is somewhat rounded and centrally placed only when your section has gone through the nucleus. If it has gone through the tapering end, obviously cell will not show presence of nucleus. Remember to draw the connective tissue supporting them and the connective tissue enclosing them into fascicles. Of course, remember also to draw the capillaries. Let's label this now. I've drawn my TS here so that you can compare it with the LS for further understanding. So here you are seeing the myocyte C. When the section went through the center, the nucleus is C. When the center went through the tapering end of the smooth muscle, you will find empty looking cells. Nucleus is rounded and somewhat centrally placed. Connective tissue sheath is present here as well, helping to convert the smooth muscle into fascicles, and here are blood capillaries. I've given you this table also comparing the skeletal and smooth muscle. Pause the video to read this, please. I hope this has helped you to draw in your histology journal as well as helps you to draw in your university examinations. Thank you, students. See you in the next Draw and Label video. Bye.